Hello everyone, I've posted a couple documents here to help you out with your final food safety project that you'll be submitting on Sunday. The first one is a sampling plan. We have the guidance notes on the sampling plan. So it's just going to help to explain the sampling plan that you have to do. So for the sampling plan, there's a few different parameters. So depending on what your food is going to be, there's going to be different types of tests that you're going to want to perform, different types of organisms you're going to try and grow and test for. So let's just go to a random one real fast. Okay, so then this is for meats, different types of meats. So let's say if you have a boneless meat, beef, veal, pork, or mutton, then you're going to be performing APC. This is your method reference, your case, plan class, and CM, capital M. So how these all work is it kind of explained right here, but basically N is the number of samples you have. Then you have your testing methods, which are from the BAM manual. And then you have your microbiological limits, which are a lowercase m and a capital M. So how this works is, depending on your sampling plan, you're going to have either just a lowercase m or a capital M, whether it's 2 class or 3 class. So how 2 class is going to work is you have your samples that are tested, and then if all of the samples are less than or equal to m, so they have fewer amount of organisms as what is said, so that's your microbiological limit, so you don't want to have more than this. So then you're going to accept if all of them are below that, or if they're less than or equal to C. So C is the number of samples, right, of n samples that can be greater than m. So in this case, you can have 3 samples that are greater than this number, but it gets a little bit more tricky when it gets to 3 class attributes. So now we have a lowercase m and a capital M. So in order to accept it, you need all the samples to be either less than or equal to m, or less than or equal to C of n samples are within greater than m and less than or equal to the capital M. So it needs to be, so you can have a number of samples that are greater than m, but they have to be less than or equal to the capital M. So once they're over the capital M, then you cannot use that anymore, okay? So that's your limit. So if you can have, in this case, you can have 3 plates that are above 10 to the 6, but if it's above 10 to the 7, then that's unacceptable, okay? So you can find a little more information in here about creating these sampling plans. I want you to take a look at that if you have any questions about it. And then you can help to use this. This is just the ICMSF book 2 I posted. So take a look at this. Both of these can be found right here in the food product sampling plan. So this should help you out quite a bit with your food safety project report that's going to be due. Okay, on to lab 11. So let's take a look at this. So the big 3, what could I talk about? So these are the big 3 pathogenic microorganisms that are going to be causing food-related problems. So the first one is E. coli 157H7, which is going to be commonly found in ground beef, raw milk, juice, vegetables, and water that's been contaminated by, well, fecal matter. So what are the symptoms? Well, you're going to have some bloody diarrhea, severe abdominal pain. It's supposed to be very unpleasant, from what I've heard. So there's the Jack in the Box outbreak of 1992, 1993, that had about 4 deaths and greater than 700 infected total. And now the less severe Chipotle Mexican Grill restaurant outbreak, which happened in 2015, which only had around 60 people infected that they know of. So not nearly as severe. So if we were to look at the total amount of outbreaks, we can see some states have been hit more hard, such as California, Washington, Oregon a little bit, but nothing too crazy. So our next organism is Salmonella, which is going to be found in a lot of raw food products. So it's going to be causing abdominal cramps, diarrhea, fever, nausea, and vomiting. It can last 4 to 5 days. So the people who are going to be at most risk are people with a weaker immune system, so children under 5 years old, and then older people, they're immunocompromised individuals. So a notable outbreak of this one was milk in 1985, which had 9 deaths and 5,295 infected. And then peanut butter, which had the same amount of deaths, but fewer infections. Okay, so take a look at that. We have quite a few outbreaks with this organism, over 1 million illnesses. Okay, so next one is Listeria, which is commonly found in ready-to-eat foods, soft cheese with unpasteurized milk, so it hasn't had the organisms killed, and then refrigerated smoked seafood and dairy products. So this one, the symptoms are going to be fever, stiff neck, vomiting, some muscle weakness, and then again, newborns and pregnant women, anyone with a weakened immune system is going to be the most susceptible. So there are a couple outbreaks with queso fresco and cantaloupe, so keep those in mind. And then this one has quite a few less outbreaks, as we can see. Very, very few illnesses caused by Listeria monocytogenes, but it's still a problem because of its ability to grow in colder temperatures like the fridge. Okay, so this next part is going to talk a little bit about API strips and PCR. Okay. Okay, so part A, we have our selective plating. So this would have been using EYA, which is short for egg yolk agar, which is going to be testing for Clostridium growth, the anaerobic growth, okay? So on this plate, it's going to allow Clostridium to grow, and when it does, it's going to have white, irregular, dry colonies, and it's going to smell very rotten. So luckily we don't have to do that, because we're doing it online. And then some may have white precipitated zones, and if that's the case, then they're going to be positive for lecthinase. And then if they have a clear zone around them, that's because of the proteolytic properties. So they're able to break down the proteins. And then some also might have a pearl sheen on the surface, and that means that they're positive for lipase, okay? So for E. coli 157H7, we'd be growing them on 2 different things. We have our sorbitol McConkey agar, SMAC, and our E. coli 157H7 biochrome agar. So they're going to be looking a little bit different. With the sorbitol McConkey, the E. coli 157H7 will have no color, whereas the normal E. coli will be pink because of the sorbitol difference. So E. coli is positive, 157H7 E. coli is negative. And then for 157H7 biochrome agar, it contains MUG, which is going to be testing for or reacting with beta-glucuronidase. So since E. coli 157H7 is beta-glucuronidase negative, it's going to be pink, whereas other E. coli would be blue, okay? That's what makes it this biochrome agar, because it looks at different colors, or that's why it's called that. So the next one is Salmonella. So we're going to be using a hectoin enteric agar, which is going to cause it to appear blue to blue-green, and then there is a chance that it could have black centers, and that's just due to the H2S production. And then we also have biochrome agar. Just like the E. coli, it's going to be a different color depending on whether it's salmonella or not salmonella. So if it's salmonella, it's going to be mauve or dark purple, and then if it's non-salmonella, it may be blue or other colors. And so those are the only ones I really need you to know about, but there are other tests that you can do, such as BSA or XLD, but we're not going to be using them or testing you on them. So you always want to be using multiple tests. So if all of your tests show positive, so including your antibody test and everything, then it's a pretty good assumption that it is indeed salmonella, but you want to confirm it with other biochemical and molecular methods. Okay, so the next organism is Listeria. So for that, we use the biochrome Listeria plate. So for L monocytogenes, Listeria monocytogenes, it's going to be blue-green, and then it's going to be surrounded by a white and gray halo. Whereas the other Listeria species will also be blue-green, but there's not going to be a halo around them. So that's how you differentiate them, just the presence of this halo or not. So for part B, for API strips, just depending, this is basically a system of biochemical tests, which allows you to identify an organism. So these different names, API 20E, API 20NE, API 20NE, or just 20NE, are going to be testing for different types of organisms. So they just have different biochemical tests inside that pack. So if you're going to be, when you use your EMB plate and you streak it, if you get purple colonies with black centers or a metallic sheen, then you have coliforms or you have enteric bacteria. So then you'd use the API 20E for enteric. And then if you have no to weak growth on or pink neutral color on your EMB, then that means you have no enteric bacteria, and you're going to be doing the API 20NE for non-enteric. And then the very last one is 20NE, which is for oxidase positive organisms. Okay, so spore formers, if it's aerobic, then bacillus, then it's probably you're going to want to use the B series selective auger to differentiate at that point. And if it's anaerobic, then it'll be Clostridium, but will not show up on APC results. So for non-spore formers, if it's catalase negative, then it's lactic acid bacteria. If it's catalase positive, then Carinibacterium. So you're going to be doing, you would be doing the API Carini. And gram-positive cocci is catalase negative. So if you have a gram-positive cocci and then it is catalase negative, then it's probably going to be a lactic acid bacteria. Whereas if it's catalase positive, then it's probably going to be a staphylococci or micrococcus. So from there, you'd be doing the Baird-Parker, biochrome staph, and a coagulase test, and then the API staph. Okay, so make sure to watch these videos on inoculation and reading results. They'll be very helpful. And then, so once you've prepared your inoculum, then you're going to suspend the colony in the medium, and you compare that to a standard, which is called the McFarland standard, which is going to be used to test the turbidity and compare it. So you know that you have one McFarland standard that is, so it has a number, and the higher the number, the more turbid the solution is, the McFarland solution. So then you compare that to the solution you make. So you suspend your colony in the liquid, and then you want it to be the same amount of turbidity as the McFarland standard that you're comparing it to. So for example, if you look at the table on the right, you have Carini, then you want it to be 6 McFarland. So you get the McFarland tube that is 6, labeled 6, and then it's going to be a certain amount of turbidity. And then you want to add your colonies to that solution until it's the same amount of turbidity as the McFarland solution that you're using, or it says to compare it to. Okay, so these are all just the instructions for API strips, but we're not going to be doing any of that. So for part C, the last one is detection of salmonella by PCR, or polymerase chain reaction. So this is going to be used to amplify and detect specific genes. So the components of PCR, you have your DNA template, which is your sample, and then you have your primers, which are going to be specific to the gene that you're interested in, the one that you're testing for. So in this case, since we're testing for salmonella, we're going to be amplifying the INV-A1 gene, okay? Then it's also going to have your Taq DNA polymerase, which is going to put everything together. Then you have your nucleotides, which are like the building blocks, and then you have your reaction buffer, okay? That'll provide a suitable chemical environment. Okay, so we have 3 different steps of PCR. You have your denaturation, which is going to denature and break apart into single strands. to make it available for the primers to attach, which is going to take place in the annealing step, which is where the primer binds. And then you have your extension, which is going to extend the primers using Taq polymerase and the nucleotides. Okay, so we'll do that 25 to 35 cycles, and at that point, the DNA will be amplified over 1,000,000-fold. Okay, so here's just a picture of the cycle. Okay, then here's your pipetting. Accuracy, not a huge deal. And then when you're adjusting the pipettement volume, make sure to keep everything in mind. So we have P20, P200, and P1000. So depending on which one you're using, it's going to be a different number. You have to keep that in mind. So yeah, that's it. Good luck. Make sure to finish the quiz and the project. And with that, I'll see you next week. Where we have our final.